Hi, we shall discuss on segmentation and the anterior posterior body plan in Drosophila. And the key concepts which we are going to discuss include zygotic genes, the protein gradients in the early embryo, bicoid as the anterior morphogen, localization of bicoid mRNA, localization of nanos mRNA, and gradients of specific translational inhibitors. There are a hierarchy of genes which establish the anterior-posterior polarity in Drosophila, and they divide the embryo into specific number of segments with different identities. And this hierarchy is initiated by the maternal effect genes that produce mRNAs that are placed in different regions of the egg. And these messages encode transcriptional and translational regulatory proteins which diffuse through the syncytial blastoderm and activate or represses the expression of certain zygotic genes. And the first such zygotic genes to be expressed are called gap genes, and they are called so because mutations in them cause gaps in the segmentation pattern. And these gap genes, they encode transcription factors, and differing combinations and concentration of these gap gene proteins regulate the transcription of another set of genes called pair rule genes, which divides the embryo into periodic units. The transcription of the different pair rule genes result in a stripped pattern of 7 transverse bands perpendicular to the anterior-posterior axis. The proteins encoded by these pair rule genes, they are transcription factors that activate another set of zygotic genes called segment polarity genes, whose mRNA and protein products divide the embryo into 14 segment wide units, establishing the periodicity of the embryo. The protein products of these gap genes, pair rule genes, and the segment polarity genes, they interact together to regulate another class of genes called the homeotic selector genes, whose transcription determines the developmental fate of each segment. And this is a generalized model of Drosophila anterior-posterior pattern formation. Here, the picture numbered as 1 is the maternal effect genes, which can be considered as the master controlling genes. It can control the zygotic genes, of which the first one is the gap gene, and the next one is the pair rule genes, followed by the segment polarity genes. And the protein products of these gap genes, pair rule genes, and the segment polarity genes can together control the homeotic selector gene. And next comes the protein gradients in the early embryo. And when we discuss about these protein gradients, there arises 3 questions. If there were gradients, what were the morphogens whose concentrations changed over space? And the next question is, what were the genes that shaped these gradients? And did these morphogens act by activating or inhibiting certain genes in the areas where they were concentrated? The answer for these 3 questions is given by a 3 set of genes. The first set of genes which encoded morphogens for the anterior part of the embryo, and another set of genes encoded morphogens responsible for organizing the posterior region of the embryo, and the third set of genes encoded proteins that produce the terminal regions at both ends of the embryo. The 2 maternal mRNAs are the bicoid and the nanos, which are critical for the formation of the anterior-posterior axis. The bicoid mRNAs, they are located at the anterior tip of the unfertilized egg, and the nanos mRNAs, they are located at the posterior tip of the unfertilized egg. The bicoid mRNA is shown in red colored region in the anterior region of the unfertilized egg, and the nanos mRNA, here it is shown in blue colored color in the posterior region of the embryo, in the egg. And these distributions occur as a result of the dramatic polarization of the microtubule networks in the developing oocyte. After ovulation and fertilization, the bicode and the nanos mRNAs are translated into proteins that can diffuse in the syncytial blastoderm, forming gradients that are critical for the anterior-posterior patterning. And this table shows the three set of genes, the anterior set of genes, which include the bicode, experientia, swallow, etc. The posterior set of genes include the nanos, caudal, Oscar, etc. And the terminal set of genes include the torso-like genes, etc. And coming to the anterior morphogen, which is the bicode. The bicode, it was the head morphogen of the Drosophila and was demonstrated by the find it, lose it, move it experimentation scheme by various scientists. And the first one was the find it experiment, in which bicode protein was found in the found in a gradient highest in the anterior region. And the next is the lose it experiment, where the embryos lacking bicode mRNAs could not form a proper head region. And the next is the move it experiment, where the bicode mRNA was moved from a normal embryo to a bicode deficient embryo. And the place where the bicode mRNA was injected became the head region. Moreover, the areas around the site of the bicode injection, it became the thorax. And when injected into the anterior of the bicode deficient embryo, the bicode mRNA rescued the embryos and they developed a normal anterior polarity. And moreover, any location in an embryo where the bicode message was injected became the head. If bicode mRNA was injected into the center of an embryo, the middle region became the head, with regions on either side of it becoming the thorax structures. If a large amount of bicode mRNA was injected into the posterior end of a wild type of embryo, it resulted in the formation of two heads, one at either end. And this is the diagrammatic representation of all these experiments. The left one shows the normal development, in which a wild type embryo which already possessed a bicode mRNA resulted in the formation of a normal wild type phenotype with the acron at the anterior terminal region, followed by the head, followed by thorax, followed by abdomen, and finally the tail region with telson. And the next diagram, the second one, shows the development of a bicode deficient mutant, that is BCD minus. It results in the formation of an embryo with two tails at both anterior and posterior ends and abdomen in the center. And the next three experiments, the first one is the injection of the bicode mRNA to a bicode deficient embryo, to the anterior region of the bicode deficient embryo. When it already lacked the bicode mRNA, and if we are adding a bicode mRNA to the bicode lacking embryo, it results in the formation of the normal head, acron region with head, thorax, abdomen, and telson with head and tail, and results in a normal development. That is, the bicode mRNA rescues the embryo from the embryo and result in the formation of a normal embryo. And if you are injecting the bicode mRNA to a bicode to the center region of a bicode deficient embryo, it result in the formation of head region in the middle region and with both tails at both ends. And if you are adding bicode mRNA to a normal wild type embryo to the posterior region, then it results in the formation of an additional head in the posterior region, resulting in a head-tail-head pattern, that is, with two heads and tail in the center. These were the three experiments, find it, move it, and lose it experiment by the set of scientists. Next is the localization of the bicode mRNA and the anterior pole of the oocyte. The 3' untranslated region, that is the UTR region of the bicoid mRNA, it contains certain specific sequences which are critical for its localization at the anterior pole. And these sequences interact with the exponentia and swallow proteins while the messages are still in the nerve cells of the egg chamber. This bicoid protein complex, it is transported out of the nerve cells and into the oocyte via microtubules. And the complex seems to ride on a kinesin ATPase, which is critical for taking the bicoid message into the oocyte. Once inside the oocyte, this bicoid mRNA complex, it attaches to the dynein proteins that are maintained at the microtubule organizing center at the anterior region of the oocyte. Thus, the bicoid mRNA gets localized in the anterior pole of the oocyte itself. Next is the localization of nanos mRNA in the posterior pole of the oocyte. The posterior organizing center is defined by the activities of the nanos gene, and the nanos message becomes bound to the cytoskeleton in the posterior region of the egg through its 3' UTR region and its association with the products of several other genes like OSCAR, STOFN, etc. If nanos or any other of these maternal effect genes is absent in the mother, no abdomen forms in the embryo. And we'll go more deeper into the mRNA localization, nanos mRNA localization in the posterior pole of the oocyte. The nanos mRNA, it usually gets trapped with the OSCAR protein and moves to the posterior pole of the oocyte or the embryo. Here, normally what happens is that the OSCAR mRNA, it binds with the STOFN mRNA, and with the help of a kinesin ATPase from the nerve cells, these mRNAs are transported to the posterior end via the microtubules. And upon reaching the posterior region, this OSCAR mRNA are translated by the STOFN into OSCAR protein, which then binds with the nanos mRNA. And thus, this nanos mRNA reaches the posterior region of the oocyte. But the most of the nanos mRNAs gets not trapped and reach the posterior end. Rather, it binds with another protein called smoke and cup protein, CUP cup protein. That what here happens is that the smoke protein binds to the 3' UTR region of the nanos mRNA, and after binding to this 3' UTR region of the nanos mRNA, the smoke protein recruits another protein called CUP cup protein. As long as this cup protein is bound to the 3' UTR region of nanos mRNA and smoke, it cannot pass through the ribosomes and get translated. And at the completion of oogenesis, the bicoid mRNA, it is anchored at the anterior end of the oocyte, and the nanos mRNA is tethered to the posterior end. And these mRNAs, they are dormant until ovulation and fertilization, at which time they are translated. Since the bicoid and nanos protein products are not bound to the cytoskeleton, they are diffused toward the middle regions of the early embryo, creating two opposing gradients that establish the anterior-posterior polarity of the embryo. And next comes the gradients of specific translational inhibitors. There are two more maternally provided mRNAs called hunchback and caudal, which are critical for the patterning of anterior-posterior regions of the Drosophila body plan respectively. And these two mRNAs, they are synthesized by the nerve cells of the ovary and are transported to the oocyte, where they are distributed ubiquitously throughout the syncytial blastoder. But if they are not localized, how do they mediate the localized patterning activities? The answer is that it turns out that the translation of the hunchback and caudal mRNAs is repressed by the diffusion gradients of nanos and bicoid proteins respectively. That is, at the anterior region, the bicoid protein prevents the translation of caudal mRNA. How it happens is that the bicoid, it bounds to the caudal's 3' UTR region and binds to a protein called BIN3, which stabilizes an inhibitory complex that prevents the binding of mRNA 5' cap to the ribosome, so that the 5' region of the mRNA cannot pass through the ribosome and get translated into the protein. And by recruiting this translational inhibitor, the bicoid prevents the translation of caudal in the anterior region of the embryo. That is, at the anterior region, the caudal proteins cannot get translated. And what happens in the posterior region is that the nanos protein, it prevents the translation of hunchback mRNA. That is, the nanos in the posterior region of the embryo, it forms a complex with proteins called pumilo and BRAT. And this complex binds to the 3' UTR region of the hunchback mRNA, where it recruits another protein called D4EHP and prevents the hunchback message from attaching to the ribosomes. That is, the hunchback protein cannot get translated in the posterior region of the embryo. And what happens in the terminal region is that the boundaries of the acron and telson are defined by a protein product called torsogene, and they also get translated only in the terminal regions of the embryo, both terminal ends of the embryo. The result of these interactions is the creation of 4 maternal protein gradients in the early embryo. That is, an anterior to posterior gradient of bicoid protein and an anterior to posterior gradient of hunchback protein, followed by a posterior to anterior gradient of nanos and a posterior to anterior gradient of caudal protein. And these two graphs shows the model of anterior-posterior pattern generation in Drosophila. The first graph is the pattern in the oocyte. That is, the bicoid nanos and the hunchback caudal mRNAs are deposited in the oocyte by the ovarian nerve cells, and the bicoid message, they get sequestrated in the anterior region, and the nanos message, it is localized to the posterior pole of the embryo of the oocyte. And the hunchback mRNA and caudal mRNA, they are ubiquitously present throughout the anterior to the posterior region. But after cleavage, that is after fertilization, the bicoid protein gradient extends from anterior to posterior, whereas the nanos protein gradient extends from posterior to anterior. The nanos here inhibits the translation of the hunchback message in the protein posterior region, whereas the bicoid protein, which prevents the translation of the caudal message in the anterior region. And this is how the anterior-posterior patterning occurs in Drosophila. Thank you.